So, welcome back to our class. This is the continuation of the cell organelles that we have been dealing in the last class. So, today we are going to learn about lysosomes. So, as we all know, lysosomes are discovered by Christian de Douai in 1950. It is a bag of enzymes which are known as acid hydrolysis. So, the macrophages are rich in lysosomes. So this is a typical structure of lysosome. It has got a membrane, then the hydrolytic enzyme is inside the center, and then there is protein, membrane protein, transport proteins, and it is covered by a lipid layer. There is a lipid layer, then there are membrane transport proteins, and the hydrolytic enzyme, that is the acid hydrolysis. So the endocytic vesicles and the phagolysosomes are fused with the lysosome to form a secondary lysosome or a digestive vacuole where the foreign bodies gets digested. When the lysosomal membrane rupture, it will release the hydrolytic enzyme causing tissue damage. So what happens is that the phagolysosomes gets fused with lysosome to form a secondary lysosome. So secondary lysosome or a digestive vacuole. So the foreign substances gets digested there. So when the lysosomal membrane rupture, it will release the hydrolytic enzymes causing the tissue damage. And the marker enzyme of this lysosomes is known as the cathepsin. So in the previous slides also we were discussing what a marker enzyme is. So what are marker enzymes? Marker enzymes are those enzymes that are specific for a particular cell organelle. So for cell membrane we learned 5' nucleotidase. For mitochondria we learned ATP synthase, malate dehydrogenase. Likewise, for lysosomes the marker enzyme is cathepsin. So this is what happens. There is phagocytosis, there is a large particle, and the large particle gets bound or engulfed by this during the process of phagocytosis to form a vacuole where the bag of enzymes, that is the hydrolysis, act, and there will be destruction of the foreign particle. So the main functions of lysosomes include the destruction of foreign materials like bacteria, viruses, etc. that enter the cells, and it also hydrolyzes the complexes like carbohydrates, proteins, lipids, and nucleic acids. So the main function of lysosome is to destroy the foreign materials that enter the cell. So we know it is a bag of hydrolytic enzymes which helps in destruction of the foreign materials. So during the process of phagocytosis by the lysosome, there is a rapid release of reactive oxygen species like H2O2 and oxygen, hydroxide, where when they come in contact with the bacteria. So this phenomenon is called the respiratory burst or the oxidative burst. It plays an important role in the immune system. So during the process of phagocytosis by the lysosome, there will be release of reactive oxygen species. Look, phagolysosome, there is formation of reactive oxygen species like superoxide and hydrogen peroxide, which when comes in contact with the bacteria, there will be the process of destruction will occur, and that process of destruction is called the respiratory burst. So next organelle is the tight junctions. So tight junctions means these are the connections that will hold together two cells. So it is a specialized membrane structures when two cells are in close proximity. So these tight junctions will permit the movement of substances across the cells through the small hydrophilic pores, and some sort of communications also occurs between these cells. So at certain positions, the subepithelial spaces of organs they will seal off, tight junctions will seal off subepithelial spaces of the organs from the lumen, and they contain specialized proteins known as occludins, clodins, and also adhesion molecules like cadherins, catenins, and vinculins. So these are the tight junctions that makes the what movement of substances or the hydrophilic small molecules, the exchange of small molecules will take place through these tight junctions. So the epithelial cells are very tight, they're tightly bound together, and these are tightly bound together by means of these tight junctions. So next is a gap junction. It is popularly called as nexus. These are the channels on the lateral surface of adjacent cells through which the molecules are exchanged. So gap junction is also called the nexus, and these are channels on the lateral surface of the adjacent cells through which the molecules are exchanged. So coming to the last topic in cell, that is the transport across the cell membrane. So we know many substances get transported across the membrane. It can be micromolecules or macromolecules. So it is through passive diffusion and active transport that micromolecules are transported, and the macromolecules are transported mainly by endocytosis and exocytosis. So then coming to the transfer of micromolecules, that is under passive diffusion comes simple diffusion. We have learned in our smaller classes that diffusion is the flow from a higher concentration to a lower concentration. So here also the solute will flow from a higher concentration to a lower concentration. And then the second one is facilitated diffusion, where the solute flow is from higher concentration to a lower concentration, but by means of what? A protein transporter. So passive diffusion is of two types, simple diffusion and facilitated. Simple diffusion is just the flow of solute from a high concentration to a lower concentration. And in facilitated diffusion, the solute flow is from a higher to a lower concentration, but by means of a protein transporter. So there is someone who will help the transport of these substances. And then comes the active transport, and these are commonly known as the pumps. That is the movement of solute across a membrane against the concentration gradient, and this is a energy consuming process, or this requires the release of energy. So that is the sodium potassium ATPase pump is the most common example that we are considering in case of the active transport. In this thing, what happens is that for 3 sodium that is extruded out, 2 potassium is taken in with the hydrolysis of ATP. So this is the diagram of the sodium potassium pump, where you can see through the hydrolysis of ATP to ADP and phosphorus, therefore the exclusion of 3 sodium, 2 potassium is pumped into the cell. 3 sodium out and 2 potassium into the cell occurs. So this is the movement across the plasma membrane, that is the passive transport and the active transport. And through the passive transport, it is of 3 types: osmosis, diffusion, and facilitated transport. And active transport, vesicles and through protein pumps. And we have learned the example of a protein pump, that is the sodium potassium ATPase. And then in vesicle comes the endocytosis and exocytosis. And thus we finish our topic on cell. Thank you.